This conference will now be recorded.
Yeah.
Very good morning to one and all. So you can at least interact with me in the chat box. I am Dr. Narottam from Dehradun, DNA Laboratories, a Center for Applied Sciences. So I think it's already 10. तो मैं थोड़ा सा वेट कर रहा था कि बच्चे और जुड़े हमसे, क्योंकि इसमें काफ़ी सीनियर फैकल्टी मेंबर्स भी जुड़े हैं, डॉक्टर्स भी जुड़े हैं और कई प्रोफेसर्स और एकेडमिशियंस भी जुड़े हैं और काफ़ी स्टूडेंट्स जो बॉटनी जूलॉजी से वो भी जुड़े हैं. So it is a humble request from my side to Dr. Nitesh, sir. Can you please mute your mic? It is a humble request, Dr. Nitesh. आपका शायद माइक ऑन है, आप उसको म्यूट कर दें ताकि सभी लोगों को प्रॉपरली सुनाई दे सके. तो मैं रिक्वेस्ट करूंगा डॉक्टर नितेश से कि वो अपना माइक म्यूट कर दें. Okay, so because some of our students, those who are in BSc itself, first of all, I request all the participants to mute their mic. Otherwise, कई बच्चों को नहीं सुनाई देगा. तो मैं चाहता हूँ कि आप अपना माइक म्यूट कर दे, please. Right. So today, basically the BIO setup, I came to know that some of the students have taken a very important initiative in Bihar as well as in Jharkhand state also. They are working very hard and they come up with this BIO setup, a very important name, I guess, because the name which they have at least taken is very important, and it is now up to them like how they are going to at least take this legacy continuing in the upcoming days also. So first of all, on behalf of DNA Laboratories, a Center for Applied Sciences, Dehradun, I welcome one and all here in this webinar. So this webinar is going to tell something about the basic job opportunities to the students, as well as like, because it is a sort of the interactive sessions with the students also. We will be taking their queries also, and we will be having some other guest members also. So it is a very important, in fact, the session which is going to happen. So a very good morning to all. I got a message from Dr. Amod Kumar sir. He's the assistant professor in TNB College, Department of Biotechnology. I welcome you, sir, here in this platform. Shivam and Aragyadeep, so they requested to us that we should be having some sort of interactive session for the students, right? So basically this DNA Labs, Center for Applied Sciences, it deals with molecular diagnostics, right? So I will be telling you about the molecular diagnostic, a very prominent field for you student also in the upcoming days, how you can make your career in these fields also, and how you can opt the different strategies or the different things or the different laboratories. So we will be talking in this presentation. So again, I request from Dr. Nitesh to at least mute the mic, please. Dr. Nitesh. Dr. Nikesh, sorry, Dr. Nikesh, can you please mute your mic because it is causing disturbance? It's a humble request from you. If you can at least mute your mic, Dr. Nikesh. Okay, so welcome to one and all. So basically, this early to means in means late 2019, and in this 2020, like it is a pandemic of this COVID-19 disease due to SARS coronavirus 2, and it has shut down so many doors as well as so many things like there some of the job opportunities it has been shut it down. So I will be taking a very relevant and very important things for the students, like how we can proceed further for this one, how you can at least opt the things that is very relevant to you during the lockdown as well as post lockdown also. So career opportunities for the biological science, career opportunities in the field of virology, career opportunities in agriculture. There are so many things where you can prepare yourself. So I think in TMB also one webinar was there where I presented some of the slides. I think it will be common for you people, like what make a very important ideal job, or some of the students or some of the people, they might be looking for the entrepreneurships, right? So means we can have a very important ideal job when we are with some of the things that may be common. One is skills, आपको काम आना चाहिए, ठीक है? ऐसी जॉब्स या ऐसा काम या ऐसा एंटरप्रेन्योरशिप होना चाहिए, या तो एंटरप्रेन्योरशिप बनने से पहले आप कुछ एक एक्सपीरियंस ले लो, या अगर आपको किसी जॉब में जाना है तो उसका ट्रेनिंग ले लो, उसके लिए सीखो. Ideal jobs is it is depending upon the three important factors: skills, जो आपको काम आता है, and what is your interest. It is not like it is going to have a burden on you. Fine. So you have to at least take care of your interest also, that yeah, this is my interest and I am going to have this thing in my future also. Skills, you should know what are the things that you knows, right? And then your interest, what is your interest, how you are going to at least customize your skills into the interest, that is again very very important. Then your values of the market, because whenever it is a demand in the market, then also your demand is increased, right? So you have to at least see like what is basically the demand that is existing in the market. That is very very relevant also to have a very good job. Otherwise, like if you are doing the job only for the sake of the title or only to earn the money, then after some time exhaustion will be there. After some time you will be bugged up. After some time you will lose interest. After some time you will lose the skills, or after even some time you will not love what actually you will be doing. So it will be a total burden. So there are n number of things that you can do in your life, and it depends upon how you are taking care of yourselves, right? So right track is very very important. Fine, because there are so many things that is existing in the market nowadays: bioinformatics, biotechnology, patents, diagnostic research, vaccines, cloning, genetic engineering. There are so many lucrative words, there are so many fancy words right now, but what actually you want and what is actually inside you, because you are the most important person to decide where you want to go. That is important. So your goals should be set in a way because you know what is your strength and what is your weakness. So the most important decision maker is you itself, right? So I think students, am I audible and screen you can see? If yes, you can put in the chat box as a yes so that I can proceed further. Am I audible to one and all? So we will proceed. Okay, thank you, Shivam, Aragyadeep. They are doing good work in for the sake of the students also, because it is a very important thing to take care of the students as a student itself also. It is a very demanding field. This is how basically I started my journey also. I will tell you like how started my journey and how we established the different laboratories too. ओके सो गोल्स, आपको राइट ट्रैक चूज करना है. बहुत सारे फैंसी वर्ड्स आएंगे और सभी. नॉलेज इज वेरी वेरी गुड, दैट इज वेरी वेरी फाइन. नॉलेज कभी भी वेस्ट नहीं होती है, बट when it comes to at least make your career, to cater your career, to customize your career, right, to tailoring your career. So it is a very important to choose what is your skills and interest. On a single track you go if you want to at least prepare your path, right? So again, this is important, because in Dehradun itself, whenever the students are coming from all across the country, so I used to at least tell them to like how their interest is, in which field they're interested. So you can see like career in research. Those students, those people who want to go into the research fields, because Corona, this virus has opened so many opportunities nowadays in therapeutic. It is there in discovery research, because new drugs, new formulations, these are the companies. I will tell you like what are the various companies in the upcoming presentation that I have already shared this presentation to so many students in our group as well as in Bihar and Jharkhand too. So in discovery research, you can see like preclinical research where there is the trials. Fine, say for example, some sort of the therapeutic hormones or some sort of the drug is being manufactured by n number of companies. So there will be preclinical trials or preclinical research. There your demand is there. Then pharmaceutical and product development, your demand is there. Project management, some of the projects, laboratories, it's there, because we have started a e-laboratory. I will tell you the concept of the e-labs also, like how we are developing the students in this lockdown through live demonstration and online e-laboratory live practical demonstration to the students. Then clinical development, some of the drugs, some of the vaccines, some of the injections, some of the therapeutically relevant products are being prepared. Then regulatory affairs, it is again very important. Say for example, there is a kit of sugar, very simple, or some sort of like say, ELISA kit is there. Now it is coming into India
Okay,
and it
okay.
will be going market, right? So before going into the market, because it should be validated for in vitro diagnostics, that is called
Right.
IVD. Sumit, can you please mute your mic? Sumit, Sumit Kumar, please mute your mic. Thank you very much. Regulatory affairs, it is again very important. अगर आप देखोगे नेशनल इंस्टीट्यूट ऑफ बायोलॉजिकल नोएडा में है, ठीक है, वहाँ भी आप जॉब के लिए जा सकते हो और वहाँ भी देख सकते हो इन नोएडा एंड ऑल रेगुलेटरी अफेयर्स का पूरा जो गवर्नमेंट का इंस्टीट्यूट है, इट इज कॉल्ड एज NIB, that is National Institute for Biological Sciences, NIB basically, right? So what actually they used to give is they used to give the approvals to the kits. They used to give the approval to the diagnostics kits, some sort of the sensor, biosensor. Say for example, you have prepared a real-time PCR kits or the PCR kits for the diagnostic purpose, but you want this license to at least sell and to make it feasible or make it applicable in the market, right? So for that thing, some sort of approvals you will be requiring, and that is given by regulatory affairs people, because there are so many types of the jobs, students, for you. It is not only the laboratory associated jobs, it is also the jobs that is there in the market also. You have to click those one. Regulatory affairs and medical affairs, this is the same thing. Marketing, कई बच्चों को घूमना पसंद है, लेकिन अभी तो लॉकडाउन है बिकॉज ऑफ दिस कोरोना वायरस. It is there, but still, because some of the companies they are hiring the biotechnologist and microbiologist, biochemist for the sales and marketing, right? So it is again very important. मैं आपको कुछ एक एग्जांपल बताऊंगा अभी. कुछ कंपनी के नाम बता रहा हूँ मैं अभी, जैसे रॉश है, काइडन है, जिमित्रा है, ट्यूलिप है, मोल बायो है, राइट? इसी प्रेजेंटेशन में आगे सबके नाम दिए हैं. ट्रांसेशिया है, बायोरेड है, जॉनसन एंड जॉनसन है, राइट? एंड दिस सिस्मेक्स है और बैकमैन कुल्टर है. और ये सारे के सारे जो कंपनीज मैं गिना रहा हूँ, ये हायर करते हैं बायोटेक्नोलॉजिस्ट, बायोकेमिस्ट, माइक्रोबायोलॉजिस्ट को फॉर सेल्स एंड मार्केटिंग एंड विद गुड रेलेवेंट पैकेज ऑल्सो. ऐसा नहीं है, अच्छा पैसा भी देते हैं बच्चों को. फाइन, सो you can opt those job also if you are not interested in the lab work, but you want to stay in your field only, right? You don't want to go into and apply in the banks and all, but you want to at least be in your setup, in your platforms only, in your areas only. So it is better to at least have all these sort of the jobs. Product support, अगर आपने मान लो महावीर कैंसर रिसर्च इंस्टीट्यूट पटना में आपने एक सिक्वेंसर बेच दिया किसी कंपनी ने, तो उस सिक्वेंसर को चलाने के लिए, उसके आर्टिफैक्ट्स को ठीक करने के लिए, उसको प्रॉपर सब कुछ उसका सही तरीके से चले उन लोगों के पास, डॉक्टर्स या साइंटिस्ट के पास, वो कंपनी हायर करती है प्रोडक्ट स्पेशलिस्ट या एप्लीकेशन साइंटिस्ट. So applications ये 1-1.5 करोड़ का मशीन होता है. So company also hire some of the people that will take care of their product support, that will support those things, right? It is not like for example Samsung ने अपना प्रोडक्ट बेच दिया तो उनका सर्विस खत्म हो गया. वैसे ही हमारा वाला जो फील्ड है उसमें भी प्रोडक्ट सपोर्ट चाहिए, right? Because when you were working in the research institute, big research institute, and also you can see like means crores rupees of the instruments. So companies used to hire those product specialists or those product scientists that will take care of all these things. Corporate communications. This is basically the backup supports that the laboratory gets from the different companies. Business setup, business development, operation and manufacturing, जो manufacturing वगैरह करते हैं, and this quality controls. Quality controls matters everywhere, right? Because अभी ई laboratories में हम सिखा रहे हैं बच्चों को how to set up the labs. एक एक चीज, छोटी छोटी चीज को लेके कि how to at least set up the quality based laboratories. What is NABH? What are the different things that you should be having? Then bio ID and all, right? So Kothagatu Vandana, can you please mute your mic? Okay, so I have already elaborated all the things to you. So these are basically the various companies or the recruitment things that is important for you people. I will share this presentation to you, right? Okay, again, this is very, very important because there are BSc students and PG students, even research scholars also, because nowadays it is not like the naukri type of the job that everyone is searching कि मुझे job ही चाहिए, ठीक है? Some of the people have started their own work, and some of the universities and this college, they are in collaboration with the industries how to at least formulate a certain kinds of the products, right? With respect to the funds or the grants that they are getting from the different sources, and how they are connecting to the industries, how from lab to land concept is very important. अगर आप एक फार्मर को अच्छा बायो फर्टिलाइजर दोगे अकेडमी से, दैट इज यूनिवर्सिटी से, और उसको कोलैबोरेट करा दोगे इंडस्ट्री से, so automatically it is going to help our farmers also. So it is basically the need that you are fulfilling, as well as how it is going to get collaborated with academia to the industries, that is laboratory to land, or it is also called as bench to bedside, from your laboratory to everyone. Fine. So again, it is a very important thing because we believe in all those stuffs, because some of the products we prepared in the laboratory and we marketed to the Johnson and Johnson and J Mitra. They're the big firms. They also require the research-based industries or research-based peoples that can come into the market, and this one. Fine. So students, networking is very, very important, because more your network is related to your fields, more will be the chances of getting the jobs, right? So you have to make your own network, informational interviews, you can see how to apply, career fairs, recruiters, you have to at least make your network. So we have to build our network, where आपके seniors है, कोई scientist है, कोई प्रोफेसर्स है, उनसे जुड़ो, राइट? ये देखो कि क्या क्या चल रहा है, कौन सी लैब में क्या काम हो रहा है, आपके लिए क्या रिक्वायरमेंट है, क्या आप उसकी रिक्वायरमेंट फुल कर पा रहे हो नहीं, राइट? सो नेटवर्किंग इज वेरी वेरी इंपॉर्टेंट, राइट? बिकॉज वो आपको रेफर करते हैं आगे काम करने के, राइट? एंड लोकल प्लेसेस, आप ये वेबसाइट्स पे अपना आप अकाउंट बना सकते हो क्योंकि आपका फेसबुक और इस पे तो अकाउंट होगा ही, बट व्हेन वी टॉक अबाउट द प्रोफेशनल थिंग, right? So baybiotechreview.com, these are free sites where so much relevant data related to the laboratory based work or related to any sort of the work uh, you can see, right? So this is again very LinkedIn, सभी को पता है, इस पे आप कनेक्ट हो सकते हो, इसमें हमें पता चलता है कि कहाँ कहाँ वैकेंसीज है, कहाँ कहाँ क्या हो रहा है, right? So this is again very important things for us to at least develop. Fine, it is not only the Facebook from where you can have the gathering or where you can make the lasagning, right? It is also very important to at least connect with the professional people, right? So professional people is like in LinkedIn, and this slide I have already shown to you. Fine, so you can prepare the network, means after MSCs and all in this one, and start searching a very good jobs. पता नहीं कब कौन से लोग आपको मिल जाए और आपके प्रोग्रेस में हेल्पफुल हो. So I am showing all these one how you can have. Fine. So resume development, ये भी बहुत important चीज है. How to at least prepare your resume. तो बच्चों resume भी आपको बनाना आना चाहिए. Resume, what are the contents you are keeping in the resume, that is very very important. एंड रिज्यूमे के साथ साथ यू शुड पुट कवर लेटर, राइट? कवर लेटर का मतलब होता है कि आप किस जॉब के लिए अप्लाई कर रहे हो, कहाँ से आपको एडवर्टाइजमेंट का पता चला, कहाँ के लिए अप्लाई कर रहे हो और आपका स्किल्स क्या है, राइट? से फॉर एग्जांपल देयर इज अ वैकेंसी ऑफ मॉलिक्यूलर oncologist in Mahavir Cancer Research Institute, and you got to know that in the newspaper of yesterday only, it was the advertisement, right? So you can write down to the project investigator at Mahavir Cancer Research Institute, Patna. Subject: applying for the position of molecular biologist at your esteemed institute, right? This is Shivam Pandey from this college. I have completed this from this college with this much grades, and I came to know that with advertisement of this one published in newspaper on this date, I am applying for the job of this one, right? Attaching my resume with this covering letter, right? Then it become very lucrative things so that it can please the interviewees also. तो ये बहुत ही इंपॉर्टेंट है. यहाँ एक्चुअली डीएनए लैब में हम अभी एक साइंटिफिक पर्सनालिटी डेवलपमेंट पे बहुत काम कर रहे हैं कि कैसे प्रेजेंट करवाएं बच्चों को, कैसे वो कर सकते हैं काम. तो ये छोटी छोटी चीजें बहुत इंपॉर्टेंट है कि आप कैसे प्रेजेंट करते हो. ये सारी फील्ड्स आपको पता है कि क्लासिकल बायोटेक्नोलॉजी, मेडिकल बायोटेक्नोलॉजी, हाउ these are the various fields where you can go. Fine. So इन क्वालिटी कंट्रोल्स, लाइक क्वालिटी कंट्रोल इंस्पेक्टर्स होते हैं फार्मा में, एनालिस्ट होते हैं, इंजीनियर होते हैं, सेफ्टी स्पेशलिस्ट होते हैं, सेफ्टी मैनेजर्स होते हैं, सुपरवाइजर्स होते हैं, देन क्वालिटी कंट्रोल मैनेजर्स होते हैं एंड डायरेक्टर्स होते हैं. बिकॉज से फॉर एग्जांपल यू आर वर्किंग इन हॉस्पिटल, सो हॉस्पिटल क्वालिटी मैनेजर्स होगा वहाँ पर, ठीक है, और बायो वेस्ट मैनेजर होगा, बायो वेस्ट मेडिकल सॉरी बायो इंजीनियरिंग का भी एक मैनेजर होगा. सो इट हैज अ हायरार्की ऑफ लाइक हाउ द थिंग्स इज हैपनिंग, ठीक है. मैन्युफैक्चरिंग और प्रोडक्शन में इस तरह से बहुत सारी जॉब्स है जहां आप काम कर सकते हो. मैन्युफैक्चरिंग टेक्नीशियंस है, ऐसे एनालिस्ट है, देन दिस इंस्ट्रूमेंट्स एंड कैलिब्रेशन टेक्नोलॉजीज आर देयर, राइट? कि किसी का इंस्ट्रूमेंटेशन या उसका कैलिब्रेशन करना है, उसके लिए आईएसओ सर्टिफाइड कंपनीज बहुत है जहां आप काम कर सकते हो. ये फिजिक्स जो करते हैं, केमिस्ट्री जो करते हैं, उनके लिए बहुत अच्छा जॉब्स है, राइट? एंड मैन्युफैक्चरिंग एंड प्रोडक्ट प्रोसेस डेवलपमेंट साइंटिस्ट है, ठीक है, पायलट मैनेजर्स है, फैसिलिटीज मैनेजर है, मैन्युफैक्चरिंग मैनेजर है, directing of the manager. So these are basically the higher position. First is you have to learn the things what actually you want. Fine. Okay, so now basically I will be talking on the current scenario, right? Because this is again a very important thing when we talk about the molecular diagnostics. फाइन. सो मॉलिक्यूलर डायग्नोस्टिक्स का ही मतलब होता है कि आप डीएनए और आरएनए पे काम कर रहे हो या कोई मॉलिक्यूल्स पे काम कर रहे हो, उसके बेसिस पे डिजीज को बता रहे हो. सिंपल, दैट इज मॉलिक्यूलर diagnostics, right? So basically in molecular diagnostics, because you can see like in current time also, when we talk about the coronavirus and all, this is molecular diagnostic that is happening in every laboratory. So in every laboratory, with the help of molecular diagnostics itself, it is the need of the techniques, technologies, and good laboratory scientists. That's what actually we are preparing in the e-laboratory. आजकल मैं यही काम कर रहा हूँ कि how we can develop the future scientists, especially so that they can learn छोटी छोटी चीजें. Fine. So molecular diagnostics, immunological diagnostic, DNA diagnostic methods, and biosensors. इसमें आपके कहाँ कहाँ jobs है, क्या क्या है, right? So okay, when we talk about diagnostics, because virus is the first important thing, क्योंकि virus को culture करना बहुत difficult होता है. कई ऐसे viruses है जिसके लिए human cell lines चाहिए होती है. कई ऐसे viruses है जो कम से कम 3-4 हफ्ते में आएंगे culture, right? That's why culturing of the viruses are very difficult, और बहुत सारी जगह labs नहीं होती है virus को handle करने के लिए. Fine. So in those cases, it becomes very, very relevant to at least develop or to at least utilize these technologies, PCR-based technology, real-time PCR-based technology, RT-PCR-based technologies, right? So, Varsha, can you mute your mic, bachche? Varsha. So please mute your mic. Those who are coming, thank you very much, Varsha. Okay, so this is basically the things that you can take as a career opportunities. I will be talking about how this molecular diagnostics has changed the trend of diagnostic industries, and also given the opportunity for biotechnologists, microbiologists to come in this era. पहले सिर्फ डॉक्टर पैथोलॉजी वाले होते थे जो साइनिंग अथॉरिटी होते थे. अगर आप आज की डेट में NABL के लॉज पढ़े तो PhD बायोटेक्नोलॉजी, PhD मेडिकल माइक्रोबायोलॉजी, PhD मेडिकल बायोटेक्नोलॉजी, PhD मेडिकल बायोकेमिस्ट्री, they can be the signing authority and they can go into the medical fields also, medical colleges also. Varsha, please can you mute your mic? It's a humble request. Varsha. Okay, thank you very much. Thank you for the cooperation. Thank you. Okay, so the success of modern medicine depends upon the detection of various specific molecules, right? It's bacterial, fungal, parasitics. तो ये molecular diagnostics से कर रहे हैं, right? अब मैं आपको बताता हूँ कौन कौन से steps में molecular diagnostic techniques use होती है, कहाँ कहाँ आपका कैरियर है. सबसे पहले देखो, is the baby healthy? सबसे पहले, right? प्रीनेटल टेस्टिंग, जहां पर आप क्लिनफेल्टर सिंड्रोम है, डाउन सिंड्रोम्स है, टर्नर सिंड्रोम्स है, यू कैन स्क्रीन आउट. genetic testing, and you know, in Bihar means I think one of the very advanced state, I guess, because you know वो पहले वाली पिक्चर नहीं रही है, because you know Bihar is my second home also, so वह पहले वाला पिक्चर नहीं रहा है. So much advancements in the field of molecular diagnostics and maximum installation of mole bio instrument, it's in Bihar itself, right? अगर आप देखोगे मोल बायो कंपनी है एक ट्यूलिप की, और मेक इन इंडिया प्रोजेक्ट में है, कम से कम 100 इंस्टॉलेशन बिहार में है, अलग अलग जगह. ठीक है, तो सब कहाँ कहाँ मॉलिक्यूलर डायग्नोस्टिक्स यूज होती है और किस किस के लिए यूज होती है. देखो ये सारे के सारे आपके एप्लीकेशंस और जॉब अपॉर्चुनिटी ही है. इज द बेबी हेल्दी? वेरी सिंपल, अभी वो बच्चा जन्मा नहीं है, फाइन, अभी वो मदर के वॉर्म में है. सो हाउ वी कैन स्क्रीन आउट द एनोमलीज, एनेप्लॉइडीज, सिंड्रोम्स, prenatal testing, genetic testing, karyotyping, chromosomal abnormalities we can see. ठीक है, उसके बाद बहुत सारी ऐसी डिजीज होती है कि हमें पता करना है कि उसको स्क्रीन आउट करना है, इस पेशेंट में ये बीमारी है या नहीं है. दैट इज व्हाट डिजीज इज दिस पेशेंट एट रिस्क ऑफ. फाइन, से फॉर एग्जांपल एवरी इंडिविजुअल कैन नॉट हैव HIV, right? That is ruling out disease predisposition. We are screening out the disease, right? So for viral load, this HIV type 1, type 2, that is serotyping, we are detecting. There are so many diseases: chlamydia trachomatis, neisseria gonorrhea, sexual transmitted disease, right? कि इवन आपके ब्लड बैंक में देखो, अगर आप ब्लड डोनेट करते हो तो जरूरी नहीं है कि वो पॉजिटिव चीजें आए. 99% चांसेस है आप उसके लिए नेगेटिव होगे. सो वी आर रूलिंग आउट the pathogens, we are ruling out or screening out the pathogens. So what disease is this patient as risk about? Molecular diagnostics ने पूरा का पूरा कम से कम मुझे लगता है आज की डेट में 50 to 60% has replaced this thing. So it is a very good thing for you people. Has the patient a disease, अगर उस पेशेंट को डिजीज है तो उसको डिटेक्ट करना है, फाइन। उसके बाद है ड्रग सिलेक्शन। अगर आप सीवी नेट है, कार्ट्रेज बेस्ड न्यूक्लिक एसिड एम्पलीफिकेशन टेक्नोलॉजी है, जीन एक्सपर्ट है, ये मैं कंपनी के नाम बोल रहा हूँ जो आपके काम आएंगे जॉब्स के लिए। सीबी नेट हो गया, जीन एक्सपर्ट हो गया, सिफिड हो गया। वो क्या करते हैं कि मॉलिक्यूलर मेथड से ही पता कर देंगे कि ये ड्रग्स काम कर रही है या नहीं कर रही है। आप टीबी का ही केस ले लो, ट्यूबरक्लोसिस का ही, आपके स्टेट्स में आरएनटीसीपी सेंटर होंगे, रिवाइज नेशनल ट्यूबरक्लोसिस कंट्रोल प्रोग्राम सेंटर होंगे, वहाँ इंस्ट्रूमेंट्स लगे हैं। आरएनटीसीपी सेंटर में आपके लिए जॉब्स हो सकती है माइक्रोबायोलॉजिस्ट और बायोटेक्नोलॉजिस्ट के लिए, फाइन। सो drug selection, we are selecting the drugs. Even say for example स्वाइन फ्लू आ गया, स्वाइन फ्लू के केस में अगर पॉजिटिव आ गया तो उसको टेमी फ्लू देते हैं, ठीक है। तो वो ड्रग सिलेक्शन तब तक डॉक्टर नहीं कह सकता जब तक आपकी रियल टाइम पीसीआर की रिपोर्ट नहीं आएगी। टेमी फ्लू, ओस्टियोमेलिविर, ये ड्रग्स है स्वाइन फ्लू के लिए। देखो अभी पर्टिकुलर ड्रग कोरोना वायरस के लिए नहीं आई है, हम हाइड्रोक्लोरोक्विनिन और कॉम्बिनेशन ऑफ एंटीबायोटिक्स जो सिलेक्शन कर रहे हैं वो भी एक पॉजिटिव रिपोर्ट आने के बाद ही देते हैं। मल्टीपल ड्रग रेजिस्टेंस ट्यूबरक्लोसिस, एक्सटेंसिवली ड्रग रेजिस्टेंस ट्यूबरक्लोसिस, so many things. It is only and only possible for the doctors to give the treatment further on the basis of your report, means your molecular virologist or molecular biotechnologist. So it is a very relevant field for you, fine. So हर्पीस में ले लो, जिनको मिर्गी के दौरे पड़ते हैं उनके लिए आपका एसाइक्लोवीर स्टार्ट होता है। हर्पीस ऐसी बीमारी है, अगर पॉजिटिव आ गया तो उसका ट्रीटमेंट ही डिफरेंट है। So it is basically a viral detection by molecular methods that will be responsible for the selection of a drug, particular drugs for the candidate, right? So because we have already started all these things in the DNA labs also, and because I was basically in charge of, I started in medical college, one of a very important lab, Central Molecular Research Lab in SGRR Medical College, where we have done all the works. उसके बाद आ जाता है रिकरेंस मॉनिटरिंग। कई बार बीमारी ऐसा तो नहीं है दोबारा वापस आ गई है, राइट? तो उसके लिए भी हम ये सारी चीजें देते हैं, ठीक है, ये टेक्नोलॉजीज यूज करते हैं, राइट? सो कैरेक्टरिस्टिक्स डिटेक्शन, किसी भी टेक्नोलॉजीज की अगर आप डायग्नोस्टिक की बात करें, अलाइजा है, आपका पीसीआर है, रियल टाइम पीसीआर है, केमिल्यूमिनेसेंस है, सेल काउंटर है, बायोकेमिस्ट्री एनालाइजर है, अगर मशीन है तो उसका बहुत इंपॉर्टेंट चीज है। आप क्या देखोगे, कल आप अपनी लैब स्टार्ट करोगे, 10 कंपनी वाले आपके पास आएंगे इंस्ट्रूमेंट बेचने की, हाँ भैया हमारा लो, तो you have to at least think like sensitivity, specificity, and very easy to handle, right? Minimum amount of analyte that instrument can detect, उसको sensitivity बोलते हैं। How much that instrument is specific for a particular target, specificity होगी। Whether you can handle it properly or not, बहुत ही मतलब ठीक है, रिसर्च में आपके 10 दिन लग जाते हैं, कॉलेज में पता चलो उसके बाद भी रिजल्ट नहीं आता है, that is different thing. But when we are answerable to the patient, so it become very important. Okay, ठीक है, right? So a good detection system should be having 3 important qualities: sensitivity, specificity, and simplicity, right? Sensitivity, छोटे कम से कम मॉलिक्यूल्स। देखो आपके पास 3 पेशेंट है, एक पेशेंट में सिर्फ 3 वायरस है कोरोना के, ठीक है, और दूसरे पेशेंट में 4000 है, और तीसरे पेशेंट में 4 करोड़ है। आपका मशीन ऐसा होना चाहिए कि तीनों के तीनों पिक करे। ऐसा नहीं है कि आप जिस पेशेंट में 3 वायरस है, राइट, उसी को ही उसको पिक नहीं कर रहा है। दैट इज द लिमिटेशन ऑफ सेंसिटिविटी, because that is also a patient and minimum amount of the target than that an individual can be having. So this is basically the platforms or the job opportunities or the different applications you can have. Genetic testing, high throughput testing for genetic disorders, including single nucleotide polymorphism, factor 2, factor 5, CFTR, these are molecular prenatal diagnostics, non-invasive detection of fetal disease, Down syndrome, cystic fibrosis, बहुत सारे ऑन्कोलॉजी में है आपके, ये जो ऑन्कोलॉजी पे काम करते हैं, कैंसर रिसर्च इंस्टीट्यूट आपके स्टेट में तो सबसे बड़ा कैंसर रिसर्च इंस्टीट्यूट है महावीर कैंसर रिसर्च इंस्टीट्यूट, वहाँ पर ये सारी टेक्नोलॉजीज हो रही है, अर्ली डायग्नोस्टिक्स ऑफ द कैंसर्स लाइक chronic myeloid leukemia, blood cancer and all. ट्रांसप्लांटेशन मेडिसिन, ऑर्गन ट्रांसप्लांटेशंस के लिए भी जो काफ़ी सारे पैथोजेंस को हम स्क्रीन आउट करते हैं, जैसे साइटोमेगेलो वायरस है, एप्स्टीन बायो वायरस है, तो दैट इज ट्रांसप्लांटेशन मेडिसिन ऑल्सो. सो इन दिस इन्फेक्शियस जो इन्फेक्शन कॉज करते हैं, HIV, HBV, HCV, herpes, human papilloma, swine flu H1N1, bird flu H5N1, avian flu H3N2, then your novel coronavirus that is SARS कोरोना वायरस एंड ऑल, राइट. तो ये बहुत सारी फील्ड्स है जहां आप काम कर सकते हो और जो ऑलरेडी काम कर रहे हैं बच्चों, राइट. तो so we can have the say for example if I am talking about tuberculosis, टीबी जिसको हिंदी में तपेदिक बोलते हैं, कई बच्चों को पता नहीं होता है कि ट्यूबरक्लोसिस को हिंदी में क्या बोलते हैं. रॉबर्ट कॉच, the famous microbiologist, discovered this bacteria in 1884. इसको कॉच postulates भी बोलते हैं. अगर आप लेबोरेटरी में काम कर रहे होंगे तो इसको अगर पीसीआर भेजते हैं इसके लिए लैब से या कोई पेशेंट या डॉक्टर, जिसको कॉच पीसीआर भी बोलते हैं, KOCH, कॉच पीसीआर मीन्स फॉर ट्यूबरक्लोसिस, राइट. एसिड फास्ट बेसिलाई स्टेनिंग भी करते हैं हम इसमें. तो ट्यूबरक्लोसिस के लिए आप देखो, इट इज अ वेरी स्लो ग्रोइंग बैक्टीरिया, राइट, एंड इट इज अ वेरी इंपॉर्टेंट बैक्टीरिया. यू नो, हम सिर्फ कोरोना की बात कर रहे हैं अभी, क्योंकि कई सारे ऐसे पैथोजन हैं जो स्लोली ग्रो करते हैं और हमारी बॉडी को वीक करते हैं, और उसके बाद हमें इतना सिम्टोमेटिक कर लेते हैं कि रिकवर होना बड़ा डिफिकल्ट हो जाता है. तो ट्यूबरक्लोसिस में ऐसे ही होता है. इट इज अ ग्राम पॉजिटिव एरोबिक रॉड शेप बैक्टीरिया and it is basically responsible for tuberculosis, right. So you can see like these are the molecular markers, क्योंकि बहुत सारे members है tuberculosis, mycobacterium tuberculosis, mycobacterium avium, mycobacterium canasi, mycobacterium avium complex, mycobacterium gonardae, ठीक है. जो मैंने अभी एग्जांपल दिए थे आपको सफर्ट के और जीन एक्सपर्ट के, ये पूरा का पूरा उसमें इसको स्क्रीन आउट करते हैं, ठीक है. अब इंडिया में क्या हो रहा है, जितने भी RNTCP सेंटर है उसमें CB नेट लगा है, तो ये आपके लिए अपॉर्चुनिटी है कि किस तरह से आप इसको यूटिलाइज कर सकते हैं फर्दर आगे पढ़ने के लिए, राइट. सो दिस इज बेसिकली इन इंडियन स्ट्रेन्स. अगर मैं इंडिया की बात करूं तो जो हमारा माइकोबैक्टीरियम ट्यूबरक्लोसिस है उस बहुत ही इम्पोर्टेंट MPV64 जीन एंड IS6110 जीन है. आप एक्चुअली काफ़ी सारे रिसर्च पब्लिकेशंस हमने इस पे किए हैं, आप गूगल स्कॉलर पबमेड में खोल सकते हो, राइट. So these are the very relevant strain that is prevalent in Indian population and the important gene that is present in the genome of Mycobacterium tuberculosis complexes, mannose binding protein 64 gene and insertion sequence 6110 gene, right. So why I'm giving all these things to you is that because applicability of your job should also be there. It should be very specific so that you can learn, like whenever you are choosing where to go, so you should be knowing the status of the profile of that laboratory or that institute. कैंसर्स में अगर हम बात करें, फीमेल में ये सर्वाइकल कैंसर होता है, राइट. जीवांगी अनिरुद्ध, प्लीज humble request to mute your mic. Right. So cervical cancer, so many types of cancer are there: colon cancer, rectal cancer, breast cancer, cervical cancer, mouth cancer, ileal cancer, right. So means you will not believe that for all the cancers, molecular diagnostics has come up with so many ranges, right. So when you go in the different departments or in the different oncology departments, so you will find out that KRAS, this JAK-STAT assays, BRCA1, BRCA2, these are the molecular markers to screen out the different kinds of the cancers, right. So this human papilloma virus is basically a virus that is responsible for cervical cancer. Actually एक ही दिन में हम सारे टॉपिक कवर नहीं कर सकते, सब हम ई लैबोरेटरीज में वी आर टेकिंग केयर ऑफ ईच एंड एवरी एस्पेक्ट्स इन अ डिटेल लाइक हाउ वी कैन डू, राइट। तो आपने देखा होगा किसी को मस्सा हो जाता है, ठीक है, हाथ और उसमें, ठीक है, जेनाइटल वॉर्ट्स हो जाते हैं, सर्वाइकल कैंसर तक हो जाता है, दैट इज बिकॉज ऑफ द ह्यूमन पैपिलोमा वायरस, राइट। सो दिस इज अगेन वेरी वेरी इंपॉर्टेंट टू हैव ऑल दिस स्टफ, राइट। तो ये जो मैं आपको स्लाइड दिखा रहा हूँ, अभी एक दो बच्चे हैं और वो हमसे जुड़े थे, एक्चुअली मैं पहले दिल्ली में और लैब्स में काम करता था। तो एक बच्चा है अभिषेक, मेरा स्टूडेंट ही है वो, एंड ही हैड सेट अप हिज लेबोरेटरी इन पटना, एम्प्लीकॉर नाम से है। और मैं इतना खुश हुआ क्योंकि बिकॉज ही लर्नड ऑल दिस थिंग्स प्रॉपरली एंड ही इज विद हिज ओन लेबोरेटरी ओवर देयर, एम्प्लीकॉर करके नाम है, एंड ही इज अर्निंग, दैट इज अ वेरी इंपॉर्टेंट थिंग। एंड वेन एवर लाइक ही वांट एनी सजेशन, अगर मेरे पास कुछ ऐसा होता है तो मैं उसको हमेशा शेयर करता हूँ कि आप इस तरह से कर सकते हो, इस तरह से कर सकते हो, बिकॉज knowledge sharing is like you are doing a very good job. That is very very important. So this is again very important molecular diagnostics test which we can have in the laboratories. तो ये सारे के सारे कम से कम अब तो 60-70 मॉलिकुलर डायग्नोस्टिक टेस्ट आ गए जो रूटीन करते हैं, और इसका एक बहुत बड़ा एडवांटेज है, राइट। So we will not go in details like this, how we can be having the different laboratory setups for the other things too. Fine. अभी कोरोना का थोड़ा सा मैं बताता हूँ क्योंकि काफी बच्चों ने शायद कोरोना वायरस के लिए रियल टाइम पीसीआर कैसे करते हैं, आपने देखा होगा, नेजल एंड थ्रोट स्वैब लेते हैं, ठीक है। इसको ये सारी चीजें नेट टेक्नोलॉजी के अंडर आती है। पीसीआर सबने पढ़ा है, दैट इज पीसीआर इज आल्सो एन एग्जांपल ऑफ न्यूक्लिक एसिड एम्प्लीफिकेशन टेक्नोलॉजीज, राइट। तो ये सार कोविड 2 जिसको नाम दिया गया है ये वायरस, इसके अंदर 3-4 इंपॉर्टेंट जीन्स होती है, न्यूक्लियोकैप्सिड जीन, अर्ली जीन, स्पाइक जीन एंड आरडीआरपी जीन, दैट इज आरएनए डिपेंडेंट RNA polymerase gene, right? Because SARS, that is severe acute respiratory syndrome, it was pandemic in 2002 and 2003. उसी जेनरा का है, उसी का ही एक सब मेंबर है जो अभी नोवल कोरोना वायरस है, राइट। तो इसके लिए और भी डायग्नोस्टिक मेथड है, बट द मोस्ट इम्पॉर्टेंट द रिलायबल इज रियल टाइम पीसीआर ओनली। फाइन। तो इसके लिए हम ये इसका पूरा का पूरा डायग्नोस्टिक एल्गोरिदम है, किस तरह से करते हैं, यू कैन सी। दोनों नाक से और मुंह से सैंपल लेंगे, ठीक है, उसको वायरस ट्रांसपोर्ट मीडिया में लेंगे। इट इज द रेट्रो वायरस, इट इज हेलिकल शेप स्ट्रक्चर दिस कोरोना वायरस एंड इट इज अ रेट्रो वायरस एंड वी हैव टू आइसोलेट द आरएनए, और उसके बाद हम उसमें प्रोब और प्राइमर मिक्स देके रियल टाइम पीसीआर में लगाते हैं और उसका रिजल्ट इंटरप्रिटेशन करते हैं। ये देखिए, now this is basically how actually it is happening in the whole world right now in the laboratories. This is what actually we are teaching to the students in e-laboratory programs like clinical and diagnostic virology, application of the virology in the routine diagnosis of the various infectious viruses, including your coronavirus, swine flu virus, your herpes, human papilloma, HBV, HCV, HIV, all those sort of the things how we can do. वर्ल्ड वाइड में अगर आप देखोगे अभी तो दो मीन्स हर एक लैब में, चाहे आप कोई भी लैब लो, we are they are doing with the two different types of PCR, one specific that is for novel coronavirus and one is generic that will screen out the SARS coronaviruses. Fine. So because these are the particular gene that is present in the SARS coronavirus to include gen RDRP. This is basically the open reading frame sequence. Open. B. Shivani ma'am, can you please mute your mic? B. Shivani, please mute your mic for the smooth functioning. Thank you, madam. So two setups of the PCR we are taking. One is novel coronavirus, that is recent one जो अभी चल रहा है, उसके लिए न्यूक्लियोकैप्सिड जीन और आरडीआरपी जीन लेते हैं, राइट। और इंटरनल कंट्रोल्स एक ऐसा जीन है जो हाउसकीपिंग जीन होता है, हम उसको सेटअप करते हैं because in our laboratory we are about to get this approval and already we are doing this swine flu and other viruses and all, and it is bit similar also, ऐसा नहीं क्योंकि बहुत डिफिकल्ट है ये करना। और एक हम जेनरिक, उसके लिए हम अर्ली जीन और हैक्स इंटरनल कंट्रोल लेते हैं, राइट। So different platforms of the real-time PCR you can do, because it is a retrovirus, right? RNA containing virus. So we are putting this reverse transcription first. Fine. For 5 minutes at 50 degrees centigrades for one cycle, right? Because this is basically the real-time PCR programming that you have to set up. Fine. तो ये सारी चीजें हम सिखाते हैं बच्चों को, how to at least prepare the programs, right? Then tag DNA polymerase activation. Again, it is a very important step, because reverse transcription will convert the RNA into the cDNA, because corona is retrovirus, RNA containing virus, and further once cDNA is prepared, it will be amplified, where the tag DNA polymerase will get activated for further amplification. Fine. So it is having 3 steps: denaturation, annealing, and extension or elongation. Fine. So in real time PCR we will be getting इस तरह से results आते हैं हमारे. अगर आप patient के result देखोगे आजकल, it is like this amplification curve, sigmoid curve, S-shaped curve जो दिख रहा है आपको, S-shaped curve जो दिख रहा है आपको, sigmoid curve, we will be having this one. Fine. So we can have the CT values with respect to the samples. Fine. So CT value is nothing but cycle thresholds, and this is basically the results interpretation real-time PCR. कभी हम इसको अलग से, because it will take at least 1 or 2 hours for how to set up all these things separately, but I am talking about the applications of all these things, like for the patients, how you can interpret the results, and you can give the positive results or negative results based upon the target amplified. Fine. So this is how we used to give the results. Fine. So this is corona, and this I am showing about the companies that is working in India, right? ये Indian based company है, और देखो CSIR है, ये labs तो आपके लिए है ही है, ICMR labs है, ये भी है, ICAR labs है, ये भी है, मेडिकल कॉलेज हॉस्पिटल है, ये भी है. मैं कुछ ही कंपनीज की बात कर रहा हूँ जो प्राइवेट लेबोरेटरीज है, और कुछ बहुत अच्छा स्टूडेंट्स को पैकेज भी देती है, लाइक रॉश डायग्नोस्टिक, काइजन, बैक्टन, Novartis, Abbott. Yeah, Abbott is a very good one. Hologic, Agilent, Cepheid, Siemens, bioMérieux, Dr. Lal, Genex Diagnostic, OnQuest Lab, Quest Laboratory, Max Group of companies, Fine Metropolis, Dr. Reddy, J Mitra, Genome Diagnostics, City X-ray Diagnostics, Ortiz, CSIR, ये तो सारे है ही, all pharma and medical institutes. Fine. So this is basically the market which we are having now. On a very because some of the students, they were very interested, like, sir, how and what you will be taking, and how you are taking care of the e-laboratories. Bacha, ki, because nowadays it is a lockdown. Fine. So students cannot come into the laboratories in Dehradun. They are not able to come. So what our team has done from the past 2 months, they have beautifully designed the training manuals, e-training manuals that we used to give to the students, as well as some of the journal concepts and the live demonstration from the laboratory. Say for example, आपको कोरोना वायरस टेस्टिंग सीखना है, हमारे पास रियल टाइम PCRs है, और हमारे पास एक्सट्रैक्टर्स, ये सारी laboratories है. We used to keep live from the DNA labs, as well as we have recorded more than 50 to 60 protocols in the laboratories from the past 2 to 3 months, which we used to at least show them directly. E-laboratory, I think it is DNA lab that has started this initiative to at least make the student well aware of, like, what is a very relevant in this era, as well as in post lockdown era, क्योंकि अभी हर एक मेडिकल कॉलेजेस में, हर एक हॉस्पिटल में इसकी जरूरत है. So again, that's very important, because I think the webinars कोई बच्चा बोल रहा था कि वेबिनार का इतना important नहीं है. So webinar is different thing, but in academics and all, because we have collaborated with so many colleges and universities in the form of memorandum of understanding for the practical work, which will be live from the DNA laboratories. So I will tell you a bit about this one, and then we will end up the session today. So e-laboratory, it is a initiative that the DNA lab is taking, where there will be the concepts, experiment protocols, e-practicals, and live demonstration given by the labs. And 29 अप्रैल को ये लेटर भी गवर्नमेंट ऑफ इंडिया से आया था, एचआरडी मिनिस्टर निशंक सर के उसमें निशंक पोखरियाल HRD minister in this meeting, they have said that the student can take care of their projects and dissertation as well as internship programs with the help of the e-laboratories, right? So the basic features of this thing, I will provide you all these contents if you will be needing for this one, right? Because we have started our batches also and about to start 1 or 2 batches in July and August. Fine. So live demonstration, deliberation, and conduction of the various experiments, and like those who will be interested, because we have not keep so much fees for this thing because it is online itself, and we know because of the financial burden on the student, we cannot heap up so much thing on them. Fine. So हमारे जो तीनों के तीनों ट्रेनिंग मैनुअल्स है प्लस कुछ एक प्रोटोकॉल्स है, राइट? हम पहले ही बच्चों को मेल कर देते हैं ताकि उसको देखें एक बारी, और डेली 1 घंटा या 1.5 घंटा 2 घंटा हमारा ऑनलाइन लेबोरेटरी प्लस उसमें कुछ एक थ्योरेटिकल कंपोनेंट्स रहता है जो हम काम करते हैं. फाइन. सो इन दिस वे वी आर we used to proceed. Fine. So after successful completion of the program, yeah, certificate जो आपके कॉलेज में लगता है वो देते हैं. And basically what we are focusing is to make the students scientifically fit, because it is a stress sort of the things in the student, like in 2020 it is going like T20 match. So what actually we are planning is that can we make the students scientifically developed so that in lockdown also thoda thoda wo seekh sake, and Saturday and Sunday is off, because we do not at least put so much burden on the students because they are also having the sort of other assignments too. For 1 month or for 45 days, even 3 to 4 days we are keeping in this week only, that's all. Fine. So scientific personality development, startup formulation and revival plan, that is important, because it is not only this sector that has been affected, because the agriculture sector, dairy sectors, this fishery sectors, this microbiological sectors, diagnostic sectors, it will be required, and it is requiring the good stuff or the good brains, so that some sort of the people they can start. अभी हमने एक नया स्टार्टअप प्लान भी स्टार्ट किया है, मैं उसको भी शो करूंगा आपके साथ. Like it is DNA labs is ready to provide the laboratory for any faculty, any student if they are coming up with some sort of the project. ठीक है, अगर आप कुछ ऐसा प्रोजेक्ट लिखते हो जिसमें कुछ अच्छे ऑब्जेक्टिव्स है तो हम उसको कर सकते हैं collaborations में, and it is science is all about collaborations, what I feel. Fine. So this is again very important things, and we are taking live projects. We are not doing that sort of things that is happening in the college level, right? So you can see like what actually we are teaching in the e-laboratory is, this is basically, for example, molecular characterization of the novel coronavirus, its diagnostics, its results interpretation, and its importance. So we are teaching all those sort of stuff. Swine flu, there is a project. Tuberculosis, then HBV, HCV, then herpes, hybrid capsulosis, cervical cancer, then for ankylosing, molecular characterization, and then some sort of the biochemical characterization on the vitamin D levels and all. So these are basically so many projects you can see in the project code, the different types of the projects we are taking care of, so that student can learn what is basically the demand of the industries, what is basically the demand of this. काफी सारे इंडस्ट्रियल माइक्रोबायोलॉजी के, फर्मेंटेशन टेक्नोलॉजी के, क्लिनिकल माइक्रोबायोलॉजी के भी प्रोजेक्ट्स हैं. So you can choose like what you want to do for 15 days and 1 month. We are keeping this much on also, and for 45 days and 2 months we are about to at least start some courses. So this is basically the training manuals. ये 3 मेन मैनुअल्स है जो हमने बनाए हैं. एक पूरा का पूरा मॉलिक्यूलर बायोलॉजी पे है, बायोमेडिकल, सेलुलर एंड मॉलिक्यूलर डायग्नोस्टिक्स. बहुत ही सिस्टमैटिक मैनुअल है जिसमें आपका स्टार्टिंग से बायोसेफ्टी से लेकर सारे के सारे चैप्टर्स है इसमें अबाउट मॉलिक्यूलर कैरेक्टराइजेशन. ये दूसरा हमारा इंडस्ट्रियल माइक्रोबायोलॉजी फर्मेंटेशन टेक्नोलॉजी का है। ये तीनों के तीनों मैनुअल्स हम बच्चों को देते हैं। और ये पैथोलॉजी का है, और ये अभी हम स्टार्ट करने वाले हैं। It will start, but some of the contents it is already there. Clinical and diagnostic virology, right? So here manuals, because I was designing from the past 1 year all these things, but now we have converted it for the e-laboratories. एंड इस तरह से हम, बिकॉज ऑलरेडी द बैच हैज स्टार्टेड, वेस्ट बंगाल के बच्चे हमारे पास, गुजरात से कोटा यूनिवर्सिटी है, एन कॉलेज पटना, साइंस कॉलेज पटना, सेंट्रल यूनिवर्सिटी ऑफ साउथ बिहार पटना, काफ़ी बच्चे ज्वाइन किए हैं. So after completion of the certificates, we used to give the certificates like this, so that whatever the topic you have chosen, that should be mentioned, right? तो हम properly इसको कर रहे हैं ताकि as per the UGC norms we should fulfill all the criteria, right? So I think this we have already covered. ये देखिए, आई थिंक ये मैंने बता दिया आपको कि जो गवर्नमेंट फर्म्स है आपकी और जो अप्रूव्ड ICMR स्लैब्स है, ये मैं स्लाइड्स आपको दे दूंगा, ठीक है, आरोग्यदीप को, because he is basically our now the student as well as now the collaborator. BIO setup, we will be working together because we want collaborations from the people, right? सो ये सारे के सारे इंस्टीट्यूट हर एक स्टेट में आप देख सकते हो, यहाँ यहाँ आपके जॉब अपॉर्चुनिटीज के चांसेस है. तो ये सारी प्रेजेंटेशंस आई विल शेयर to the Aragyadeep and he will give to you. There are also some of the industries. uh, yeah, we will give to the organizer, those who is organizing this one, Aragyadeep. And Aragyadeep, please share your contact number, so that means we can share all these presentation. There is no issue. I have already shared to so many students, because this I have prepared from the ICMR guidelines, like where is the availability of the job opportunities for all the youngsters or all the beginners and all. So this is the things you can see, like the vaccine production forms, क्योंकि काफ़ी सारे बच्चे चाहते हैं कि वैक्सीन प्रोडक्शन में जाएं, तो ये सारी की सारी कंपनीज वैक्सीन प्रोडक्शन में काम कर रही है. अगर ये देखें आप, भारत बायोटेक है, बायोमेड है, कैडिला इज देयर, चिरॉन इज देयर, डेनो वैक्सीन एंड बायोलॉजिकल प्राइवेट लिमिटेड इज देयर, बायोफार्मा इज देयर, पनिशिया बायोटेक, रेनबैक्सी, सीरम इंस्टीट्यूट, शांता बायोटेक, and I have given the address of these companies also: GlaxoSmithKline, then Sanofi Pasteur India Institute, and Central Research Institute, National Institute of Biological, Cipla, Cadila, Biocon, Ranbaxy, Serum Institute, NIV, National Institute of Virology, Pune. So because all these things basically we have put in the training manuals also. Those students who would love to learn from us from the DNA laboratories, they are most welcome. And this is ISO certified laboratory, and we are working in so many forms. ये स्लाइड्स सारी लैब्स की है, ये लैब मूवी भी आप यूट्यूब पे देख सकते हो. एंड किसी ने पूछा था मुझसे स्टार्टिंग में कि आप अपने बारे में बताएं. सो आई एम डॉक्टर नरोत्तम शर्मा, राइट, एंड आई स्टार्टेड my career from this Baxton Drugs and Pharmaceutical as a quality analyst. Then I was there for only for 3 to 4 months, because I was a biotechnologist. I am a biotechnologist, and then there was an interview in a very important research institutes that was under ICAR, this under foot and mouth disease virus project. uh, I was selected as a senior research fellow there, and I worked there for 1 year. Then I shifted to Delhi and in DBT projects in Oroprog laboratories, and I was a junior scientist and the training coordinator for BITP program, that is Biotech Industrial Training Program, and I worked there for the 3 years. Then I came in medical college, and in medical college we set up the whole laboratories from zero to at least it's functional. So from there itself I stayed for 8 years. And this DNA laboratories, now we have established all these wings, and we are working here in DNA laboratories from the past 1 year. And this is how basically this e-laboratories work. So it was a very systematic thing, because we used to give the schedule to the students early itself. I think it is a very unique program in India, and even from Pondicherry, Bihar, West Bengal, Gujarat, Rajasthan, students are coming. See, this is the beauty of this science driven technology, is that at home itself you can see the live presentation or the live videos of the laboratory, what is happening. So we used to give the certificates, and some of the accreditation actually I want to show, like, because in Uttarakhand I started the first biosafety level 3 lab. It is a very important laboratory for H1N1. तो ये उस टाइम के स्वास्थ्य मंत्री जी थे, and I was awarded for this one. And the first laboratory we have started for the people, and I was a part of that one only. So there are n number of things, like, because I was working in the biotechnology, microbiology, and in this field only, in my field only, and we have developed 3 to 4 laboratories from the past 8 to 10 years, especially, and developed um, so many research projects, as well as we have means customized or prepared the basics training program protocol that is actually required for the industries. So basically, these are some of the photographs I'm sharing with you, because those are 2020 में कोई moment तो नहीं मिलने वाला है. So these are the old photographs I'm showing to you. So and it was a very nice journey. And bachcho, stick to your field only. I did BSc biotechnology from Bangalore University, then MSc biotechnology from here itself, Dehradun, and then PhD medical biotechnology from a department or DBT sponsored from Banasthali University. Then I believed in this giving the things only, or I used to think that what is basic need of the society or the science, you should make the career in that field only. This is our team. जो हमारी टीम है, सबसे अच्छा हमें ये लगता है कि जो हमारे डायरेक्टर साहब है वो संस्कृत से है. ही इज फ्रॉम द संस्कृत बैकग्राउंड. जो हमारे डायरेक्टर है वो हमारे चाचा जी भी है. तो ये ऐसी टीम है जहाँ पर डिफरेंट टाइप्स का डीएनए मिलेगा आपको. So this is what actually the very uniqueness of this lab is all about. And I want, means, so many candidates can join us in the various programs or in various project, right? So Dr. S.K. Khanna sir, he's a retired scientist, as well as he worked with us. He is a retired scientist from Indian Institute of Petroleum. Then Mr. Divya Prakash Pandey, the big name. So he is a deputy director come training coordinator for us. And our team is very young, and there are microbiologists, biotechnologists, industrial microbiologists, pathologists, and all. So we all are taking care of each other, and we all are taking care of how we can move further and how we can at least cater the things. So one program we can tie up with the different medical fields also. So yeah, this is again very important, like from DNA lab itself, because in every state there is in Uttarakhand State Council for Science and Technology, under my guidance also, in a span of 2 to 3 years, 3 Young Scientist Award was backed by Central Molecular Research Laboratory. And these were MD, and basically I am a PhD biotechnologist, but these students were MBBS MD itself, and they were working under me only. I was the co-supervisor for them also. So bacho, it is a position that you can create. It doesn't matter like what is basically the eligibility of the institute. It is the requirement that you can fulfill. So so many things we have done in the past, and it is like just a starting journey. And I want so many people to invite to work with us, and BIO SETUP, Sanjeevani Biotech, and so many firms wants to join with us for all these things. So these are some of the photographs that I'm sharing with you of my journey. So for anything, I am ending up this session over here. So I am very grateful to BIO SETUP people they gave me this opportunity to at least interact with a very beautiful audience over there. And Thank you very much. And अगर आपको कांटेक्ट करना हो for any sort of thing, I will give the presentation to Aragyadeep and Shivam. ये इसके मालिक हैं बायो सेटअप के. And see, whenever young researcher or young students, they want to at least make their career, so it's also our responsibility to take care of them also, क्योंकि उनको आगे बढ़ाना है, उनको गाइड करना है. ये भी हमारी एक रिस्पॉन्सिबिलिटी होनी चाहिए, क्योंकि आत्मनिर्भर के लिए आपके पास एक ट्रैक तो होना चाहिए, कुछ एक ऑब्जेक्टिव्स तो होने चाहिए, right? So it is not only a hypothetical things that you should be having in your minds. It should be having the plans that you should be having to at least fulfill your dreams. So thank you very much. Thanks to one and all. So any queries or anything you want to ask, you can put in chat box. आपको ये सेशन कैसा लगा, whether it was fruitful or any sort of things, you can share with me.
Hello, good morning, sir.
Good morning, good morning.
Sir, can I say something about our bio setup?
Yeah, why not? It is your form, dear. You are most welcome.
Thank you so much, sir. Good morning, everyone. Myself, Argadeep Das. I am an entrepreneur and a director and founder of bio setup. Our main motto is to help in a life science background student, that is zoology, botany, biotechnology, etc. Those students who don't know about the e-learning and e-practical handling the instrument that are using in the field of research. And I'm great thank you to Dr. Narottam sir as our senior scientist of a DNA lab to support us. We are collaboration with DNA lab for learn the best practical sessions for our site, right? And and the thanks to our biosetup team, Mr. Sivam as a managing director, Mr. Amit as a CEO, Ms. Susma, Mr. Mukesh, and Mr. Aryan, and many more to support us. And also, Dr. Amos sir is also a faculty member of a TNB college, and thanks to support us. And I also thanks to all the participants who are generally giving us a crucial time to bio setup to joining our session, right? So please support, and because you are also be a part of a family member. And those who are not joining our group bio setup, and in the of the session we are sending you at the link to joining. And guys also gives us a feedback, because your feedback is helpful to us to getting more and more energetic, right? So we are also discussing our team as soon as possible. We update many more extracurricular activities in our bio setup group. And again, thank to everyone to be a part of our family member. Stay safe, stay healthy. Thank you so much, Narottam sir, for kind of regards. Please sir, continue.
Thank you very much. Thank you, dear. So thank you, everyone. So I think all the information you can take from Aragyadeep, because he is our now this taking care of this basically the e-laboratory training programs, and he will tell you like how to at least apply and all. And for another things also, apart from e-laboratories, and also we can have the different sessions in upcoming days also. We can make out the things more useful for the students, and that will be a short sort of the this thing. Okay, one student is saying, sir, is human genetic is growing field. I'm graduate from biotech and planning from MS in human genetics. Should I go on? Yeah, dear. Human genetics is a very important field, because so much thing is coming. A very important example I want to give is that all the IVFs depends upon the human genetics only. In vitro fertilizations, just cell lineaging, cell culturing, germplasms, this because infertility in case of the males and female, that is means it's growing drastically. And after this pandemic, हर जगह तो sanitizer का छिड़काव हो रहा है. So what can be the after effects of like so much sanitization? It is not useful for the upcoming things. So this is basically providing an opportunity for this emerging field, that is the genetics base, and maximum things are depending upon this genetics itself. So yes, it is basically the emerging fields also you can go. And but one important thing is that you should study other relevant fields too, because human genetics, it is a club up of this cellular, molecular, cell biology, recombinant DNA technologies. All the club of mixture of this thing is very relevant. And it will be better if you will go in the clinical sites, in this clinical sites in human genetics. Right, Ashu? Okay, so I think we are done for the today. And thank you once again. You are a very beautiful audience. So some of the senior faculty members are also here in this group. So we will keep on sharing the key contents to you. And for the e-laboratory programs, Aragyadeep is the key player who will take care of your registration and all. If you are interested, it is up to you. Right? It is not a sort of the promotional events that we have prepared for lucrative the students and to find all these things to you. So it is up to your interest whether you want to join the e-lab or not. It is totally your decisions. So thank you very much. Thanks to one and all. So anyone wants to say, he or she can open or unmute the mic and speak. Anyone or some of the respected faculty, if he or she is hearing me, so they can open the mic and at least say few words, because I don't know so much about the participants. Okay, so thank you. Stay healthy and stay safe. Thank you very much. So now I am ending this meeting and I will send the details to Aragyadeep. Thank you very much.